So good morning, good afternoon, and good evening, everyone. I hope everyone is doing good today, and I'm happy to see that we have a number of people who have managed to join in the webinar. So welcome everyone who is on board. And just to introduce myself, my name is Caroline Thuo, and I am a communicator working with the African Genetic Biocontrol Consortium. And my passion is in science communication, and that's why we have been holding a series of webinars, this being our part 2 of the webinar. And today we'll be still talking about science communication, and we are talking about, today we are tackling techniques of addressing and communicating the uncertainties associated with emerging biotechnologies. So Aruna is supposed to be our moderator for today. I'm sitting in for him, but he will be joining us shortly as we continue or go along with this webinar. So I will take you through some of the objectives of this webinar. And one of our objectives is to raise awareness on communicating genetic biocontrol technologies in Africa. Our other objective is to address the importance of community engagement and education in genetic biocontrol technologies in Africa. And this will address the challenges, impacts, gaps, and recommendations in the communication of genetic biocontrol technology. So we'll be addressing this as we move along. And also I'll be introducing to you our speakers for the day. We have Gloria Obaki and Dixon Wilson, who are our speakers for the day. So as we move along, in case you have any questions that you may need to ask, feel free to have your questions on the chat or on the Q&A segment, and we'll be sure to address it as soon as our speaker is done. So without further ado, I'm going to introduce our first speaker, who is Gloria. And Gloria is a writer, editor, communication specialist, and public relations expert. So Gloria is a dynamic communication specialist with demonstrated expertise in understanding client objectives and crafting engaging content. She has a strong background in leveraging web and social platforms. She excels in targeting audience engagement strategies. Her extensive training and achievements include specialized courses in science communication, genome editing, agricultural biotechnology, and biosafety, which has enhanced her ability to effectively communicate complex topics to diverse audiences. So currently, Gloria is serving as the head of press and protocol unit at the National Biosafety Management Agency, that is NBMA, in Abuja, Nigeria. And Gloria plays a pivotal role in coordinating internal and external communications, managing media activities, and drafting speeches for agency executives. Beyond her professional endeavors, Gloria is actively involved in volunteer service, contributing her expertise to organizations such as African Biosafety Communicators Network and the African Science Communicators Network. She also serves on the multiple committees focused on biosafety communication strategy, gene editing guidelines, and national biosafety policy development. So a bit more about Gloria. Her education, she holds a Master's of Science degree in Peace Studies and Conflict Resolution from the National Open University, Abuja, Nigeria, and is currently pursuing a PhD in Development Communication at Nasarawa State University, Kefi, and that is in Nigeria. So with that bio from our first speaker, Gloria. Gloria, you have the floor for the first topic of the day. Gloria?
Thank you so much, Carol, for that very elaborate introduction. I'd like to welcome us to this meeting. All right, so I guess I'll just go straight to share my screen. Okay. Can you all see my screen?
Yes, we can see your screen, Gloria.
Yes, we
All
can.
right, thank you. Yes, thank you very much. So my topic is I'll be talking about genetic biocontrol technologies, the role of communicators, what communicators need to do in order to properly communicate genetic biocontrol technologies. I'll just move straight to my content. Okay, all right, so I'll move straight to my content, which is first of all, introduction to genetic biocontrol technologies and the importance of effective communication, benefits of genetic biocontrol technologies, the role of communicators, communicating science to the public, engaging stakeholders, addressing misconceptions, future directions and opportunities, and of course, my conclusion. So this is basically everything I'm going to be talking about, the content of my presentation. Okay, so I'll start off with my introduction. What is genetic biocontrol technology? So genetic biocontrol technologies, this involves the use of genetic manipulation to control populations of pests or disease vectors in a targeted and environmentally friendly manner. So these technologies typically they aim to reduce reliance on traditional chemical pesticides and offer more sustainable solutions for managing agricultural pests, disease, transmission, and invasive species. Now, I'd like to talk about some of these technologies. Examples of these technologies, we have gene editing. Gene editing, I'll just talk about briefly. It involves, of course, CRISPR-Cas9 technology that enables precise modifications to the genome of target organisms, allowing for the introduction of desired traits or the disruption of essential gene. Also, we have genetic modification. Genetic modification involves the insertion of foreign genetic material into an organism's genome to confer specific traits, such as resistance to pests or diseases. We also have sterile insect technique, which is this approach involves releasing sterile insects into a target population, which mates with wild individuals and that's wild, of course, insects, and suppress reproduction, leading to population reduction. We also have RNA interface. So RNA technique can be used to silence specific genes in pests, leading to reduced fitness or mortality. Then we have gene drives. Gene drive system can spread, the system spreads desired traits through population rapidly, potentially leading to population suppression or modification. So in the long run, overall, genetic biocontrol technologies offer promising tools, very promising tools for sustainable pest management and disease control. But they also raise some concerns, which are ethical, regulatory, and environmental considerations that require careful evaluation and communication. That is where we are, where the role of communicators come in to properly communicate this, communicate the risk, communicate the benefits, communicate the challenges, communicate all that needs to be known about genetic biocontrol technologies. So now I'm going to talk about the importance of effective communication. Now, effective communication plays a very crucial role in the field of genetic biocontrol technologies for several reasons. You know, when we talk about public understanding and acceptance, genetic biocontrol technology often involves complex scientific concepts that may be unfamiliar and misunderstood by the general public. Hence, effective communication helps to bridge this gap by translating scientific jargon into accessible language and engaging
Gloria, we can't seem to hear you. So apologies, our speaker seemed to have fallen off slightly due to internet connectivity. She is getting back right now.
Thanks.
So in the meantime, in case you have any questions, feel free to put it on the chat or on the Q&A, and we can address it as we go along. I can see Gloria is getting back now. But in the meantime, please feel free to put your questions on the chat and we'll be able to address it. Gloria, are you back?
Yes, I'm back. Technical hitches along the way. Apologies for that. Okay, I'll just share my screen again.
Okay.
Yes. Yes. Okay. Okay. So I was okay. So I was talking about the importance. Can you hear me now?
Yes, Gloria.
Yeah,
Okay.
yeah.
All right. Thank you. So apologies for the network technical glitch. So I was talking about the importance of effective communication. I talked about the need for, I talked about public understanding and acceptance. I also talked about building trust and credibility. And I'll also talk about regulating ethical and navigating ethical and regulatory challenges. You know, genetic biocontrol technologies raise ethical and regulatory challenges, questions related to environmental impact, human health, and environment. Hence, effective communication facilitates dialogue among, yes, effective, facilitate dialogue among scientists, policymakers, and the public to navigate these challenges, ensuring that decisions are informed by diverse perspectives and considerations. Stakeholder engagement is very key for effective communication. You know, when stakeholders are engaged throughout the process, from initial research and development to field trials and deployment, ensuring that their concerns and perspectives are considered and addressed, it helps in the successful implementation of these technologies and strategies. Now, in the bid to address misinformation and to counter fear, effective communication is very key because genetic biocontrol technologies may be subject to misinformation, misconceptions, and fear-mongering from various sources. Hence, effective communication counters misinformation with accurate information. It educates stakeholders, it educates the public about the potential benefits and risk of these technologies and addresses fears through transparent communication and risk communication strategies. It also helps advocate for science-based policy by providing policymakers with accurate information, highlighting the potential benefits to these technologies and addressing concerns in a transparent and constructive manners. Okay, so I'm going to quickly talk about the benefits and benefits, challenges, and controversies when it comes to genetic biocontrol technologies. Because as communicators, it is very important that we know what are the benefits, what are the challenges, and what are the controversies, as this will help us to appropriately communicate. When we talk about benefits, we talk about precision targeting, reduced pesticide use, enhanced crop yield, resistance management, and environmental conservation. By reducing the need for chemical, pesticide, and promoting integrated pest management practices, genetic biocontrol technologies contribute to biodiversity conservation and the ecosystem and the health of the ecosystem. The challenges. Of course, in every technology, there are always challenges. We have ethical consideration, we have regulatory hurdles, we have public perception. How do the public perceive of this technology? The fear of long-term effects. We have intellectual property rights, as it is happening with the case of genome editing right now. Then the controversies surrounding these technologies. We also have GMO labeling, ecological risk, social justice, corporate control, and global governance. These are just some of the controversies, challenges that surround. So as communicators, it's very important that we know what we are delving into, because when we talk about genetic biocontrol technologies, they offer promising solutions for sustainable pest management and disease control, but they also raise complex ethical, regulatory, and societal challenges and controversies that require careful consideration and dialogue, the need to properly communicate. Now, what is the role of communicators? The role of communicators will have a very big role to play. Translation of complex sciences, how do we, translate the scientific jargon, like I mentioned earlier. We have to bridge the gap between the scientists and the general public by translating complex scientific concepts and technical jargon into accessible language. They help to make this, when you are able to, break it down, also to say, it helps to make genetic biocontrol technology understandable to diverse audiences, including policymakers, farmers, consumers, and of course, the media. We also communicate and create awareness. Educate and create awareness. So these are, we educate about the potential benefits, like I mentioned before, the risk and ethical consideration associated with genetic biocontrol technologies. We also raise awareness as communicators about scientific principles behind these technologies, their applications in pest management and disease control, and their application for agriculture, public health, and the environment. We also have risk communication. Of course, we communicate the risk because communication plays a crucial role in risk communication by providing accurate information about the potential risk and uncertainties, addressing the fear. There's so many audiences with different fears. So when you communicate the risk, you are able to address the fear. We also have ethical and social implication. We have advocacy and policy influence. We have public engagement and trust building. Very, very critical for science communicators in this regard. All right, so communicating science to the public. In order to communicate science to the public, you must be able to, first of all, know your audience. Who are you communicating with? Because it's when you are able to know your audience, that is when you'll be able to know how to, coin your communication, your messaging. So you have to know your audience. If you're communicating to policymakers, you know how to coin your message. If you're communicating to the farmers, if you're communicating to the media, to the general public, you know how to coin your messages. Tell compelling stories, humanize the science, take it from a human angle. You know, when you make science more relatable, relate it to the ordinary man, relate it to the farmers, make it relatable, humanize it. It makes it more compelling, it makes it more associatable. And also use clear and accessible language, visualize complex concepts, you know, foster two-way communication. As you're communicating, you are getting feedback. It helps you to know. It helps you to know if what you're communicating is properly received, and also provide context and relevance. You have to be transparent. You have to be honest, of course. So that you shouldn't make it seem as if there's something to hide in this in the science. No, make it as open as possible. Engage emotionally, humanize again. Use multiple communication channels and also collaborate with other communicators who are not scientists. Collaborate with them. Collaborate with other with the journalists, with the media. Is very, very important. Now, in engaging stakeholders, there are some basic things that we need to know. As communicators, we must be able to engage stakeholders. You identify your stakeholders. Who are the people you want to communicate with? Identify them. You map out how you want to communicate. Map out your stakeholders, map them out, how you want to come, how you want to pass the message across to them. Then you tailor your message to meet their need, because each different audiences, different stakeholders have their different needs. So you have to tailor your message. Inclusive dialogue. You communicate, you get your feedback. You listen attentively. Ensure transparency, build trust. It cannot be overemphasized. Empowerment, conflict resolution, and long-term engagement. Ensure that you engage over time. Don't stop. Communication does not have a stop where you put a full stop. No, you keep communicating from before, even when you're conceptualizing the technology, when you start the technology, in the process, all through the process of the technology. It's very, very important. Addressing misconceptions. You know, this is very key, because as communicators, we must be able to know how to address misconception. There are so many misconceptions out there. Genetic modification is unnatural. Genetic biocontrol leads to franken food. Genetic biocontrol is unpredictable. Genetic biocontrol causes genetic pollution. Genetic biocontrol is a silver bullet. You know, so many concerns out there. You need to be able to know these misconceptions, you know, because common misconception about genetic biocontrol technology can stem from a variety of factors, including lack of understanding, fear of the unknown, misinformation, you know, and all whatnot. When you talk about, for example, there are some people that say it's unnatural. There's a misconception that genetic modification is inherently unnatural and harmful to the environment. However, many genetic biocontrol methods mimic natural processes. This is a way to address that particular myth. Tell them, you know, these things happen naturally and can be used to enhance natural biological control mechanisms. So, you know, okay, for example, I'm talking about it leads to franken food. They say there's a misconception that genetically modified organisms, GMOs, produced through genetic biocontrol technology are unsafe to consume or harmful to human health. In reality, of course, GMOs undergo rigorous safety assessment and are subject to regulatory oversights to ensure their safety. So, you know, okay, when you talk about genetic biocontrol being a silver bullet, you know, some people perceive genetic biocontrol technology as a silver bullet solution to pest management and disease control without considering the need for integrated pest management strategies and the potential for resistant development. Now, in order to correct these misconceptions, you know, you have to be able to build trust in genetic biocontrol technologies. And how do you build this trust? How do you build the trust? You provide accurate information, address concerns directly, identify the concern. When you identify the concerns of the different, because farmers may have their concerns, which may be different from policymakers, which may be different from consumers, which may be different from different stakeholders. So you have to identify this misconception and address them directly. Engage stakeholders. It cannot be overemphasized. Regular engagement of stakeholders. Promote transparency. I'll say it over and over again. Demonstrate benefits. Humanize it, like I said before. Humanize the science. Make it relatable. Educate the public. Continuous education, continuous awareness, collaborate with trusted sources. Science is built on trust. It's built on verifiable evidence, not mercy, not hearsay. So when you have trusted sources, you collaborate with them. It helps address misconception of the technology. Now, as communicators, what are the future what are the potential roles for communicators in shaping the future of genetic biocontrol? Because I tell you, genetic biocontrol is technologies is moving. As science and technology keeps evolving, we keep getting new technologies, new techniques to do things. And in all of this, communicators play a critical, a very vital role in shaping the future of genetic biocontrol technologies. The role of communicators cannot be overemphasized. By facilitating dialogue, promoting understanding and fostering informed decision-making among diverse stakeholders. The role of communicators cannot be emphasized. One, education and awareness campaign. Communicators can develop and implement educational campaign to increase public awareness and understanding of genetic biocontrol technologies. These campaigns can include informational materials, public forums, workshops, and educational outreach programs aimed at different stakeholder groups. Also have stakeholder engagement. Communicators can facilitate dialogue and engagement among diverse stakeholders, including scientists, policymakers, industry representatives, farmers, consumer groups, environmental NGOs, and the public. They can organize stakeholder consultations, focus group discussions, and collaborative workshops to gather input, address concerns, and promote collaboration. Now we have media relations. Communicators can work with the media to disseminate accurate information about genetic biocontrol technologies and counteract misinformation. Because I tell you, there's a whole lot of misinformation, and communicators have a very, very big role to play. They can provide journalists with access to scientific experts, arrange interviews, and share compelling stories and case studies to increase media coverage and public understanding. Online engagement. Communicators can leverage digital platforms and social media channels to engage with diverse audiences and promote dialogue among genetic biocontrol technologies. About genetic biocontrol technologies, they can create informative content, host online discussions, and respond to inquiries and concerns in real time to reach broader audiences and foster interaction. We also have policy advocacy, where communicators can advocate for evidence-based policies and regulations that govern the development, deployment, and oversight of genetic biocontrol technologies. They can work with policymakers, advocacy groups, and industry stakeholders to promote policies that balance innovation, safety, sustainability, and societal values. We also have ethics and risk communication. Communicators can facilitate discussions about ethical, social, and environmental implications of genetic biocontrol technologies. which can, address ethical dilemmas, communicate risk transparently, and engage stakeholders in dialogue about values, priorities, and decision-making criteria. We also have capacity building and, international collaboration. Collaborate with international groups such as, of course, Genetic Biocontrol Consortium, facilitate collaboration, facilitate international, just like we're having now, and, kind of organize conferences, workshops, and so on. By fulfilling these roles effectively, communicators can contribute to shaping the future of genetic biocontrol technologies in a responsible, inclusive, and sustainable manner, ensuring that they are developed and deployed in ways that address societal needs, protect the environment, and promote public health and well-being. I would just like to conclude by saying that communicators play a multifaceted role in addressing genetic biocontrol technologies by promoting understanding, facilitating dialogue, building trust, addressing concerns, advocating for science-based policies, and empowering stakeholders to make informed decisions. We bridge the gap between science and the society. Of course, communicators serve as intermediaries between scientists and the general public and all that. Our efforts are essential for ensuring the responsible development, deployment, and acceptance of genetic biocontrol technologies that contribute to sustainable agriculture, public health, and environmental conservation. Thank you for your attention. Over to you, Carol.
Thank you, Gloria, for that very informative presentation. So I had a question, because you said about science and technology keeps evolving, and so does communication techniques. And we find that currently social media and digital communication is the next big thing. So how can communicators utilize digital communication tools, perhaps, to address uncertainties associated with emerging technologies, and at the same time still maintain trust and also legit sources of information?
Yes, thank you. Yes, I'm here. Thank you, Carol, for that question. Of course, like I said, science and technology keeps evolving, and communicators must learn to evolve with it, utilizing digital digital media for online engagement. There are so many digital platforms and social media channels that communicators can leverage on to engage with diverse audience and promote dialogue about these technologies. With the use of digital media, online media, you can create informative content, host online discussions, bring the scientists to the table, just like we are doing now. We are bridging the gap. Bring the scientists to the table. We have YouTube channels. We have various social media platforms. We have Twitter. We even have LinkedIn, where you can post science stories. You can create informative content using, like I said, credible source, using trustworthy sources. Interview scientists, talk to the scientists, host, bring them on board on webinars, online platform, host online discussions, and respond to inquiries and concerns. This will help respond to inquiries and concerns, and even reach diverse audiences, because it works in real time to reach broader audiences and foster interaction. Even when you post stories online, it can generate interactions, because when people read it, diverse audiences go through it, and it really helps address concerns. I will urge communicators to honestly leverage on online engagement. It's a very good source of communicating science. Very good one. I hope I answered your question.
Yes, you have. Thank you so much, Gloria, for that. So I will move on. If anyone has a question that we may not have answered, please feel free to put it on the chat, and Gloria will be able to address it later on. So I'll move on straight to our second speaker for the day, who will address the impact of communicating genetic biocontrol technologies in Africa, stories and case studies by journalists and scientists in Africa. And his name is Dixon Wilson. And Dixon Wilson is a principal research scientist at Ifakara Health Institute, a professor at Nelson Mandela African Institute of Science and Technology. He's also a Wellcome Trust International Intermediate Fellow and an affiliate of African Academy of Sciences and fellow of Africa Science Leadership Program as well. Wilson is also a member of WHO Prequalification Team for Assessment of Vector Control Products. And he graduated with an MSc Applied Zoology from the University of Dar es Salaam. And he comes with a wealth of knowledge and experience armed for this topic. And Dixon, please take over. And now the floor is yours to take us through our second topic.
Carla, let me share my screen.
Okay.
Perfect. Can you see my screen?
Yes. Yes, you can see your screen.
Okay. Yes, I put it in presentation mode. So thank you very much for introducing me. I guess I have like 4 to 5, 1 hour, just to share the importance of science communication with regard to genetic engineering technology, but also to share what we have been doing as a Health Institute in terms of developing technology, and a little bit of regulation landscape, opportunity and challenge that comes with the actual development of technology, especially in this part of the world, in Africa. I guess a lot of us might be asking ourselves, why are we really struggling to communicate this? I think for every new technology comes with create a lot of fear within a public domain. It's a quite a natural tendency for human being. I guess if you look at a lot of psychological theories on fear, you realize human being tends not to like what they can't predict or control. Just like uncertainty reduction. And then you can think of something like evolutionary biology, as we want to stay safe, we wouldn't want anything that we don't know what it might be causing to us. And then there is resistance to change. We tend to prefer what we know or our current state rather than something new. So it's quite normal for a lot of people to be fear, and I think that's why communication becomes critical, not only to communicate what the technology is all about, but also to make sure that people understand. If you ask me, probably 20 years down the line, we won't be, probably the level of communication around genetic biocontrol won't be as much as it is being done at the moment. People will be quite aware of it and probably to be already in the public domain by then. So I'll start with this slide that you see. So this came in, I guess, by the end of last month. It is Unitaid when they posted that the new net actually have an effect on microbial resistance. I think it looks like a minor thing, but basically I think the lighter you are meant to say the new net can actually combat insecticide resistance, not antimicrobial resistance. So if you leave aside the text, I think what is reminding us from this post on LinkedIn is there is a need on how best, even if you're a writer, it's good to cross-check what you want to communicate before you put in the public domain. And that's why the relevance of ensuring that you communicate the right information becomes important. So basically my talk is around genetic gene drive, technology development, regulation, distribution, implementation for malaria elimination in Africa. I'm sure you can still hear me, right?
Yes,
Hello?
yes, we can
Okay,
hear you.
wonderful, yeah, thanks. So who are we? So basically I'll start speaking about the project itself. So I represent, I'm a PI for a project called Transmission Zero. Transmission Zero is basically a global malaria research program working to develop innovative technology to stop malaria transmission. We are a consortium of 4 institutions, Imperial College London, Ifakara, National Institute of Medical Research, in Tanzania, and Swiss Tropical Public Health Institute. What we work on is we focus on population modification. Another form of gene drive is population suppression. And in Tanzania is where the main work at Ifakara of modification is being done in one of our sites in Bagamoyo. And we have also started looking at vector population dynamics in future potential release sites, and we work with National Institute of Medical Research, and we do that in Ukerewe Island. I will explain why we chose the island, because this is a guidance from WHO pipeline for assessing genetic modification. Someone will ask, why are we doing this, given that we have bed nets, we have other forms of controlling malaria? Why gene drives? So if you look at this slide, basically what I want to communicate is, if you look in the last 20 years, the reduction of malaria has been only 8% in terms of incidence and only 60% in terms of mortality. And someone will say, probably if you continue with what we have, we might be able maybe to achieve zero malaria. But again, if you look, the population of Africa is doubling almost every 20 years. And that's what is affecting actually the fight of malaria itself. Back in 2000, we have nearly 241 million cases. And if you look, even in 20 years later, we still got nearly the same number. It's because of the booming population that outstrip the resources and the information that we are putting in the public domain. We are still losing one child almost every 1 minute. And I've listed a lot of problems that are actually challenging our current intervention: insect resistance, drug resistance, parasite detection resistance, urban vectors, and climate change that are extending dry season, but also not on dry season, even a lot of rain that are creating a lot of breeding actors for these vectors. In terms of investment, we don't have much. If you look, the budget for malaria control in Africa, which has nearly 95% of the budget, it's only 34%, 68 up to 70% is coming from donors in the West. We have done so much in terms of putting a lot of intervention, but they're not enough. Just last year, we distributed nearly 2.9 billion nets, but this level of investment, we don't have resources to continue doing. So you see that we need some supplementary tools to add on what you already have if we want to reach eliminations. And gene drive present one of those options among other tools. I would like to touch a little bit about the regulation aspect. So we are not working in isolation or in bubbles. If you look at Gene Drive at Convention for Political Diversity and the Cartagena Protocol on biosafety is regulated as an environmental issue, but also WHO, which oversee or sort of a guardian of what is happening in terms of research at a global scale, already released a framework on how this technology should be assessed at countries that would like to work on toward that technology. African Union, which a lot of African countries are the member to that union, they have also committed their voice that this tool has a potential for eliminating malaria in Africa. You're quite seeing Africa, because malaria is just not only in Africa, but it's because over 95% of the burden is on this part of the world. But again, the jurisdiction of who wants to implement the technology or research on the technology has left to each country based on the legal landscape, but also regulation instruments that are in that particular country. So if I zoom in and look at Tanzania, as a country, we already have the law and instrument and the frameworks how this technology should be assessed and what is required at institutions that want to work on that particular technology, but also which officers that are responsible for regulating it. So if anyone wants to work, for instance, in Tanzania, as an institute required to have what Constitutional Biosafety Committee required to package dossiers, the Minister of Environment within National Biosafety Focal Point will require to look at your dossier and they're required to give you an approval before you start doing. I guess some other countries probably don't have this, but I think Tanzania, given that we've been quite on the front of even assessing genetically modified crops, that's what has given us an upper hand also to start creating these laws and regulation now best to look at this kind of technology. The other aspect that I would like to share with you is basically like the way Gloria pointed out, the test study itself. What are we doing as a program, Transmission Zero here in Tanzania? So the first speaker also alluded a little bit of what is gene drives, but basically gene drive, what it does, it works by introducing genetic elements in a preferential way within a population. The normal way of inheriting any genetic element from parent to offspring is done by what's called normal Mendelian. And that has a 50 chances of that particular gene of interest to be passed to the offspring. What gene drive does, though, is to ensure that you design a system where you have a certainty near 100% that genetic element that you desire to bring a certain effect within a population will be passed down to offspring nearly 100%. And that's why this tool is so powerful, because if it reaches at implementation stage, you have these mosquitoes that can be released, then you have assurance that whatever you're working on is likely to be equitable and scalable, and you won't need to be repeating doing it like other intervention. So there's two forms of gene drive at the moment. One is population suppression and another is population modification, which is also termed as replacement. I guess when it's come to communication, for case of population modification, which I'll speak a lot today, is I wouldn't call it replacement because we're not replacing the population. What we are doing basically is modifying the population. So all that modification is done on the background of the genetic makeup of that particular organism, in this case, mosquitoes. And what we are driving in here is actually to make sure that mosquito cannot be able to transmit malaria. So we simply introduce a very few genes that I'll explain later which has ability to stop the development of malaria parasite insect mosquitoes. So these are some of the genes. Basically, what we introduce is what is antimicrobial peptides and meganin and melitins. These are coming from Africa tree frog, and these are honeybees. What we do is we call what we have is effector line or effector molecule, and these are the molecule that actually breaks or stop the development of malaria parasite inside the mosquitoes. So you don't have to pay much attention to the details, but what we do, the carbopeptidase is a normal gene inside the mosquitoes. And what it happens, it is being hijacked to be able to explain these two genes that are inserted inside mosquitoes, the meganin and the melittin. And not all of them actually, not the entire gene is being inserted. It's just part of the gene which are inserted inside the mosquitoes. So these are only two genes and part of those genes. And what this carbopeptidase does, it hijacked in such a way that it expresses these antimicrobial peptides, which later on with guide RNA, they are being produced as proteins and then within a midgut of mosquito, and that will stop the development of the parasite itself. And that's why I emphasize in terms of communication, this is population modification, because if you look at the entire genome, you're basically introducing two genes that has less than 60 amino acids. So it's a very tiny modification that you're actually making in mosquitoes. Someone might say actually it's negligible, but it's done in such a way that its effect is quite huge in terms of enabling that mosquito not to be able to support parasite development inside. So some studies been done previously to demonstrate that this works in the lab. And what it does is what I just said, that antimicrobial peptide interferes with plasmodium development inside the vectors. And it actually stops the oocyst development, which is one of the stage of parasite development towards the infective stage, which we call sporozoite. And it's shortened the lifespan of mosquito itself. So mosquito that we modify in our lab actually have 4 days shorter compared to the wild type population, which is another advantage. So how can you test this technology step by step? This is what I was telling you. If you look here, you'll see some of these, the effector molecules, and we have what we call transmission blocking regime. And this case is actually the effector itself, which we put it inside mosquitoes. Remember, at this stage, this is not gene drive. Mosquito is gene drive. It's just that mosquito that are carrying only two genes that has ability to block the development of parasite inside. And then what we'll do, we want to test if that works. If you take the blood or the gametocyte from the population and feed it to that mosquito, will that modification stop the development? Yes, if it does, then later on we can put in the drive and assess the ability of that gene being carried from one generation to another generation. As I said, to be able to have what we call a gene drive, you need to have that modification, and you must be able to drive that modification within the population. So a mosquito that has been modified just to block the parasite is basically genetically modified mosquito or transgenic mosquito. But once you combine that with a drive, so to ensure that you can be able to carry that effect from one generation to another, then that's where it becomes gene drive mosquitoes. And for us, we are testing this on step by step. One is because they are separate component. One is effector trait that actually is, can we really make modification? Yes. Can this modification block the parasite? Yes. And then later on, we move on to see if this modification can be driven in a population. And at the moment, we are working with Northwest Gambia, which is one of the very efficient malaria vectors. Other species that we are planning, we are also working on Anopheles albiensis, and we are planning also in future to start working on Anopheles, which is a highly efficient malaria vector in a lot of countries in Sub-Saharan. As I said before, the modification that we are making is very minimal. And this is important because if you put a lot of modification inside the mosquitoes, the probability of developing resistance becomes high, because every modification that you are making has what we call fitness costs. So the minimum you are making, which is merely negligible, then the probability of that mosquito immunity to reject it becomes very negligible. In that case, the ability of developing resistance becomes. And you like to have a technology that once you introduce in the environment, you like it to do what you want it to do before the mosquito develops resistance against it. So as I said, this is vector molecule that will be tested if it has ability to block the parasite. And then once it does that, to be combined with a drive so that that modification can be driven, but also the drive itself can it drive itself. So you can compare the drive as autonomous cars, the cars without a driver, which you can program to be moving, and then you can bring in the driver to be there just to control it. So in terms of if you visualize this at field stage, what we'll do, and this map here basically portray the Okalewa Island that I pointed earlier. What we'll do first, release genetically modified mosquitoes or transgenic mosquitoes. These are mosquitoes with only effector strain that has ability to block the parasite development inside them. And at this stage, there'll be no gene drive. And basically what we're looking at is to evaluate the effector fitness and the ability to dispersion effector, the ability to move from one mosquito to another, because if it happens, it will be just based on normal Mendelian inheritance. And then you go to another stage where you want that effector, can we really drive it from just one generation to another generation? Not, let's say, from point A to point B, but not to point C. In this case, what we'll do, we'll just bring in what we call a booster. And at this stage, basically what we're evaluating the drive of the effector and the ability to block transmission. So what will happen here, once the booster is a component that's ability to drive that effect from point A to point B, but it doesn't drive itself. And then we'll go to a third stage where we release mosquito with effecting drive system. In this case, the driving system will be actually driving the modification you have done and driving itself. And at that stage, you are creating a system which becomes self-sufficient because for each generation that will be resulting from the mothers will have both the driver and the effector. And in that case, it will keep driving itself until you achieve the impact that you want to achieve at population level. And this testing is actually following the guidelines that WHO has put in public space for those who want to work on genetic engineering and genetically modified mosquitoes. So the effector molecule will be basically at small scale isolated release or confined release, which is an island. Same way the second stage, which is effector and the static source of or with a booster, will be again a small scale but open release. And then you can do at large scale open release. These are some of the, this slide basically aimed to show you that at the point where this effector molecule works, it stops the okinetic stage toward persist, and then it also have an effect on release of sporozoid by mosquito, which is an effective stage of the parasite. And you can see that from this graph, when you have a booster or when you stop, what exactly happened in terms of achieving the desired effect. This is what I explained earlier on what that we are leveraging on carbopeptidase, which is a gene inside the mosquito itself, and they're putting only two genes, meguanine and melatonin, not actually a complete gene, just part of those genes. And those genes actually with a messenger RNA, they are supposed to express protein, which is meguanine and melatonin. And this protein has antimicrobial peptide effects, which induce antimicrobial expression. And that's what you want, because its effect is stopping the parasite development, is stopping the release of oocysts, but also delayed the development of the sporozoite. And that's what it show that within the lumen of mosquitoes, the antimicrobial peptides will interfere with the parasite and then make them failure to develop to infective stage. So all this has been demonstrated in the lab, and some of these pictures in the graph actually to display is when you have mosquitoes with meguanine and melatonin carbopeptidase genes, you see that in the top panel of the picture is the oocyst is quite small, which means they didn't develop to the stage which could lead to infective stage compared to wild type, where you don't have the gene, the effector molecules, which you see the oocysts are in bigger sizes. And similar applies here in terms of looking at the sporozoite, DNA inside mosquito that have the effectors, and those that doesn't have the effectors. You reduce actually the DNA content, which means the ability of sporozoite to happen in mosquitoes by 14 to 67 times. Depends on the number of blood meal. If you have done only one blood meal or you have actually supplemented with a second one. So that was more of the to give a snapshot of the technology itself. But so in terms of progress of our program, what we have done so far in our lab, we have managed to create the transgenic mosquito. So basically, these are genetically modified mosquito with only effector strains. And what we are doing is basically to test those effector strains with parasite isolates from the human who are infected with malaria. And we are hoping that that modification will have ability to block the development of that particular parasite, which we have already just testing in the lab. And we have to make sure that we continue maintaining our colony in such a way that those modifications can last. Because remember, as I said, if this is not a modification you make and you're sure that it will always stay in mosquitoes. It's a laborious process that you continue screening even with what you have modified to make sure that modification is stayed intact and it can continue delivering the effect that you wanted to see, in this case, blocking the parasite development. All these works are done at, as I said, at Bagamoyo. We have this the container, which is our lab. So these are two sets of containers, one having all the machines that are actually powering the lab itself. And this is biosafety level 3 laboratory, which has a lot of biosecurity to make sure that there is no mosquito that can escape the plant, but also, and that is as per country regulation, but also as per WHO regulation, that this kind of work should be done in biosafety level 2 to biosafety level 3 containment facilities. And some of these works, probably some of you have seen this. April last year, we basically were the first group in Africa to create transgenic mosquitoes by Africans. And this is important in terms of communication, because I think what will guide the acceptance of technology by regulators in the public domain is showing that there is a sustainability of what we're trying to develop. But also there is a capacity on the ground of people who can actually work on this. At the moment, we're working on technology where we are creating mosquito that's ability to block the parasite development. But in future, the same scientists who have done this, they might look at another trait. Let's say they want to make some modification for mosquito that has less ability to feed on human, which means if they take so little blood, then the ability of producing eggs will be impaired. Or mosquitoes that couldn't fly a certain range, or mosquitoes that would be so small. So all this capacity creates a sustainability that our scientists has technical know-how of going beyond what they are working on at the moment. Where this facility is used not only to for our work of creating transgeners, but also what it is for our engagement and for our local communities that we are working on or collaborating with in this project. So for primary school children that are actually providing their gametocide to feed our mosquitoes have opportunity to visit our plant. So they just don't come here to offer blood, but also to learn what is happening in the facility. And then another facility. So this is another containment facility, which you call semi-field systems. So what I showed before is a pure lab, but then you have another containment facility, which sort of simulates an environment like a field environment. And the plan is once we are sure that everything that we want to do in the lab has been done quite well, then we'll be moving from the lab to the semi-field systems. In terms of regulation, each stage requires different regulation approval. Now we have approval to work in the lab. But before moving to this stage, we also request for approval to work in this system, because the level of containment in the system is not as much as in the other system. And even what you are looking at in this semi-field system, what you'll be looking at is that second stage that I showed you on having mosquitoes effector strain, and they can be supplemented with a booster, which in this case is a component that has ability to move our effector strain from point A to point B. So basically, this effect is just within one generation, not in the second generation. If you want to boost again, you have to introduce that component. If you want to boost again, you have to introduce it. And the third stage basically will be you just release once, and then it drives itself in the entire population. What we are also collaborating with other people in terms of assessing the technology itself. There's three pillars. You look at genetic efficacy. Genetic efficacy is what I've been explaining. How good your effector molecules or genetic trait that you are working on is effective, is fit. Can it stay inside the mosquitoes? Can it move? And then later on, you look at entomological outcomes. Once within the mosquito population, does it have an impact in terms of if you're working population separation in terms of reducing vector density, if you're working population modification in terms of reducing the lifespan of mosquitoes? I say that the technology we're working on can reduce mosquito lifespan, but also in terms of blocking the parasite. And then we'll go further looking at epidemiological outcome, which now is when you do surveillance within the entire population and see the impact of the technology itself in terms of malaria reduction and number of cases and mortality. So it's quite heavy slide, but it tells you different components that we have to be looking at, and it should be appreciated that there is so much that it's not as straightforward where people think someone just working in the lab and go to the field and then you measure the outcome. As I said earlier, that this is a new technology. There is so much even for us who are developing technology that we are learning as we move forward. So it's create a process of back and forth where you thought that the modification you have made is the best, and you realize at a certain generation of your modification, mosquito cannot keep it. And when you project doing that in the field, you say, no, we have to go back to square one and to design the effector so that they can be more powerful and be sustained when you go to the field. So this paper probably will come out by end of this month, so where people can be able to enjoy reading all the details. So also what I want to share, what are the opportunities is, and most of our opportunity actually apart from working in the lab, I think where we are comfortable as developers, most of the opportunity actually for us is how best to engage with regulators, but also other people working within this space, but also communicators that has a lot of influence in engaging perspective from the public, but also in ensure that the information that is being taken there is appropriate ones. So one is, if you look at side, there's a lack of contextual and technical awareness, understanding of GM and gene drive, so genetically modified and gene drive mosquitoes. And to us, this is an opportunity where, as scientists or also as a communicator, we need to make sure that within our pipeline of development, we create also a program that will be sustainable in terms of raising awareness. Second is insufficient framework to support the assessment of the submitted dossier. Remember, some of the regulators, what they've been conversant with is regulation of the technology within agriculture or other interventions. But this being new, we need, of course, them being regulators, someone might say, how best do you separate yourself from regulators and technology developer? But I think we shouldn't shy away to give the right information and the details of the technology developed. And I think by doing that, they'll have power and appropriate skill set to assess some of the application that will be submitted. At the moment, it's just one institute doing this in Tanzania. But given the relevance of this topic, we're doing research that 5 years, 10 years down the line, there'll be so many groups working this space. But the regulator will remain to be the same, and they should have capacity to regulate that. Another opportunity to leverage on the existing ethics and governance guidelines on gene editing. So we were speaking is gene drive, but the first speaker spoke about gene editing, and we know that in Tanzania at the moment, there are also a program on gene editing for sickle cell. These are all genetic modification. The difference here is just that one is just localized within one person where you're doing that editing to eliminate some rare diseases. But for our case, you are doing editing in one mosquito that you are certain that you want to be killed in another generation. And the ethics around the technology itself, actually, we can borrow from each other, and that's what I meant by leveraging existing ethics and guidelines. There's opportunities leveraging the existing technical platform, national malaria control programs, technical vector control working groups to discuss the potential of GM, gene drive mosquitoes and genetically modified mosquitoes to contribute toward malaria elimination. Quite often what happens, the discussion of different interventions within these groups or NMCPs in Africa tends to happen when the technology is already approved by WHO. But I think given the sensitivity and uniqueness of genetic engineering itself, I think it's high time when this technology should start early on within this platform, where people can have an opportunity to discuss them and even create a set of questions that they would like a developer to start looking at that in future can inform the development and fine-tuning of the technology itself. I guess this is my last slide. So if you look at Lance's report back in 2019, it predicted that by 2030, some of the technology in the public domain will be gene drive, highlighted in yellow, among other technology for malaria control. And I think as scientists in Africa, we have opportunity to doing a lot of research in collaboration with northern partners to make sure that we are aware what it takes to develop technology, what it takes to assess it, what is likely to be the question that might create fear. People might say, okay, it might affect other organisms that will feed on the modified mosquito. Can we answer those questions ourselves? So at the moment, is this research outcome that will be able to create a package for communicators to communicate to the public domain that what is being developed is safe. What is being developed is safe, but it has ABC. So the limitation and opportunity can only be packaged if research will be done. And I think we are at the stage where we have opportunity to do so. The picture on the right side is our old campus in Bagamoyo. And basically, I meant to say, when we are going to 2030, the realization of that goal can actually be done in Africa, where the burden of the disease up to 95% is actually found. So thank you very much. That's what I wanted to share for today. And I'm happy to answer any questions that will come. Back to you.
Thank you so much, Dixon, for that comprehensive presentation about the impact of communicating genetic biocontrol technologies in Africa. And also, we appreciate how much you've gone in depth in explaining some of these technologies. So thank you so much. So I will give a few minutes for anyone who may have a question. You can raise your hand or you can put your question in the chat. And Dixon or Gloria will be able to address your question. So Beth, you had asked if we will put this, share this recording. Yes, we will have this recording up on our YouTube page at African Genetic Biocontrol Consortium once the recording is done. So no pressure about that. Let me see. I can see a question from Valentine. So Valentine is saying, I'm a journalist and science communicator from Burkina Faso. Thank you to communicators for their presentation. So my question relates to communication tools, especially with social networks, how to convince populations of the importance of genetic biocontrol. In Burkina, with the example of genetically modified mosquitoes, only yesterday, there was a civil society coalition which qualified the experiment as scientific terrorism. Okay, so Dixon, would you like to take up a comment on this?
Yeah, I could give it a try, yeah, from a developer perspective. As I started saying, this is quite a new technology and it will cause a lot of discomfort within a public domain. And it is quite expected. And I think there is some mechanism on our best to deal with this. One is for a group like that can term this development of technologies is terrorism. I wouldn't separate them from the anti-GM groups. And what is more important is to appreciate their stance, and us as a developer, our responsibility is to engage with them and to give them the facts based on what we have found so far in terms of research itself. And every time we make progress, I think it's important to continue engaging them rather than seeing them on the far end or on the other side of the coin. To me, I see them as part of people who are there to shape the process itself of developing the technology. In terms of tools, I think one is, it's unfortunate that you cannot differentiate the modified mosquito versus the non-modified one if I bring you to the lab. But I think it's important people to appreciate how much has been invested in terms of safety and in terms of questions that are being addressed to decode any biological uncertainty that might come by technology development itself. So for instance, as I said, if someone is feeling that this technology might be, the modification might be passed from mosquitoes to ants that are feeding on mosquitoes, then that can be answered as a research question and data can be presented that this is not the case. If someone might say by suppressing certain mosquito population will affect the ecosystem in terms of food chain, and that can be answered by actual research itself. So in terms of tools, one is inviting them to the facility, but second is having an engagement with them in terms of detailed discussion on why would they label this technology as terrorism. So the in-depth discussion or focus group discussion to make sure that you understand which angle they're coming from and use that as opportunity to engage them. Back to you. Thanks.
Thank you, Dixon, for that answer. So we have someone saying, Gloria and Dixon, thank you for the wonderful presentation, and then goes ahead to ask a question. If you consider communication as a spectrum, how can the scientists, for example, reach out to the various stakeholders with a specific message that each group will understand without the unnecessary oversimplification that might lose context of the reports?
That's a good one. Can I give it a try? And maybe
Yes,
my
please,
colleague
go
will
ahead.
also step in. I think as a scientist, for us, this is an area where we appreciate that our skill set is not at par as what we want to go to the public domain. And I will strongly encourage for any group or organization working on this to appreciate the importance of whether consulting to communication experts or having a team member that have expertise in terms of communication. And I think one of the big part of the technology development is engagement, and engagement comes with how do you package messages for different groups. If you go to the villages where people doesn't speak English, or they have their normal living has been resonating with a certain way of defining things, then there must be a way to appreciate that culture and be able to design your message that will be easy to resonate with their culture and their understanding. And unfortunately, that's not a job of a scientist, but I think it's important for a scientist to understand what we can do and what we are not capable of, and then seek the right support from the experts in communication to guide us. So in our program, we have that. We have experts in communication that are actually overseeing all the communication. So once they map all the different groups that are existing in Tanzania at different levels, from community, from national, even at high-level stakeholders, middle-level stakeholders, community stakeholders, different groups, gauging their perspective, and then they can design different packages of messages that is on to use with them without losing the meaning. So the meaning across all the stakeholders will still be the same, but the way you communicate it will have to be different to make sure that people can understand, because that's our ultimate aim at the end, that people understand what you're communicating, regardless, without losing the meaning of that message across the screen. Back to you, Carla.
Okay, thank you, Dixon, for answering that. I see we do not have any more questions being asked from the attendees. Let me just a minute. So someone else says, thank you for the detailed presentation. Stakeholder engagement is crucial in science communication, and they are now well versed with gene drive technologies. They had no idea that they mimic natural processes. So I'm glad to see that our attendees are benefiting and learning from these webinars. So as we wrap up, a big thank you to Dixon and Gloria for being our speakers for the day. And also thank you to all our attendees for your time and for your patience through these webinars. And you kept time today, which is very much appreciated as well. So before I wrap up, finally, the Suma Sanon says, thanks to the communicators for the knowledge sharing. So indeed, what you're doing is really, really appreciated. So I will wish everyone a good morning, a good afternoon, and a good day, and a good evening as well. And we shall see you in our webinar 3 series. Yeah, and have a good rest of the day.
Thank you. Bye.
Okay, bye.